In today's lecture, we are going to talk about structure and the function of protein. In the previous lecture, we talked about the introduction of proteins and the amino acids. So today we are going to talk about how proteins work and what could be the possible classification of the protein on the various bases. So as you all know, proteins are formed by amino acids, and in general you can say amino acids are basic, structural and functional unit of proteins. And on the basis of the number and the complexity of the joining of the amino acids, proteins can be classified into the various forms. So, the first classification is on the basis of presence of polypeptide chain. On the basis of presence of polypeptide chains, proteins can be classified into two types. The first one is monomeric protein and the second one is multimeric protein. The first we will see what is monomeric protein. As we know that proteins are formed by the amino acids when the amino acids are joined together by the peptide bond, so the polypeptide chain is formed, which is called protein. So if protein is formed from single polypeptide chain, like I am going to write A to show the amino acid chain, which is a polypeptide chain. And you can also represent it like this. If the protein contains a single polypeptide chain, so such protein is called as monomeric protein. For example, insulin. And if the protein contains a number of polypeptide chains, then it is called as multimeric protein. For example, hemoglobin. Hemoglobin is the oxygen carrying protein to the blood, and it is composed of a number of or multiple polypeptide chains. So it is the multimeric protein. So this is the one way. So this is the one way on the basis of which we can classify the proteins. There are other ways also present in which we can classify the protein. Proteins can also be classified on the basis of presence or absence of amino acids. So on this base, protein has two types. The first one is complete protein and the second type is incomplete protein. So what is meant by the complete protein? As we say, it is on the basis of the presence or absence of amino acids. So complete protein is a protein in which all essential and non-essential amino acids are present. It means it is a complete protein. It has all essential as well as non-essential amino acids in it. For example, animal protein like beef, mutton, fish, or there may be any other animal protein source. So complete protein has all the essential and non-essential amino acids, and the example is animal protein. And what is meant by incomplete protein? It is a protein in which some of the amino acids, they may be essential or they may be non-essential. In which some of the amino acids are absent, so that protein is called as incomplete protein. And the example of the incomplete protein is plant-based protein. From where you get the plant-based protein? For example, pulses, vegetables, beans, etc. So it means that plant-based protein is an incomplete protein and the animal-based protein is a complete source of protein, and the youngsters need the complete source of protein by the meat, any source of meat like beef, mutton, chicken, fish, etc. And incomplete protein do not contain all essential or non-essential amino acids in it. So these are the two ways on the basis of which we can classify the proteins. Now coming towards the structure of the protein. There are some structural classification of protein. Proteins can be classified on the basis of their structure. So on the basis of their structure, proteins can be classified into the four types: primary protein, secondary protein, tertiary protein, and the fourth one is quaternary proteins. So the primary protein, if I draw a graph here, so the primary protein is the simplest protein, and as we go downward, so when we reach the quaternary protein, so it will be the most complex protein. So this classification of the structural classification of the protein is based on the complexity of the structure. Complexity of structure. Right. Okay. First we will see what is primary protein. The primary protein is the simplest protein and as you all know, the proteins are the combination of the amino acids in the form of polypeptide chain. So primary proteins are those proteins in which amino acids are joined together in a linear form. Suppose these are amino acids, they are joined together and a linear structure is formed, and this linear structured protein is called as primary protein. Okay. When the protein is formed, there is a process which is called as a translation. In the process of the translation, protein is formed, and the first when the protein is formed, it is linear in structure. It means it is a primary protein when a new protein is formed. Okay. Now here is a point to be noted that all the primary proteins are non-functional. And they become functional when they are further modified in form of secondary, tertiary or quaternary structure. So it means when a primary protein is formed, it is non-functional, and when it is further modified in the secondary, tertiary, or quaternary structure, then it becomes functional. But there is an exception case occur. Exception is insulin is primary protein and you all know what is the function of the insulin. Insulin is a functional protein, so there is a question then how it is, because it is a functional protein, it regulates sugar. It regulates sugar in blood. So it is a functional protein, and as here we said that primary protein is a non-functional protein. So here this is the exceptional case, insulin is the exceptional case, while all other primary proteins are the non-functional protein. And insulin was the first protein discovered by the scientists. Insulin was first protein discovered by scientist named F. Sanger, and it had 51 amino acids in chain. So this point is also there is also another point that all the proteins have different number of the amino acids in their structure, as the insulin has 51 amino acids in its structure. So these are the some characteristics of the primary protein. So as I said that in these forms of the proteins, we move towards a complexity of the structure of the protein. So what is the secondary protein? Proteins usually do not lie flat like the primary protein. In case of the secondary protein, primary protein turns itself and form a helical structure. And that helical structure of the protein is called the secondary protein. Right. For example, the structure may be like the linear structure turns and form a coil, and that helical structure is called as a secondary protein. And here in this coil, amino acids are present, and you all know, I am going to show the amino acids with these dots. You all know the bond between the two amino acids is a peptide bond. While in case of this helical structure, amino acids are present on this turn or on this turn too, and the linkage between the amino acid of the one turn and the amino acid of the second turn is by the H bond or hydrogen bond. The bond between the two successive amino acids is called a peptide bond, while the bond between the amino acid of the one turn of the helical structure and the amino acid of the other turn of the helical structure is hydrogen bond. So you can see that in the secondary protein or in the structure of the secondary protein, there are the two types of the bonds are present. The one type of the bond peptide bond, and the second type of the bond is hydrogen bond. And this helical structure of the protein is called as alpha helix, and this is the most common secondary structure. A secondary structure may be of another type, that is called a beta plated sheet. This would be like so. For example, this is one helical structure, this is another helical structure, two helical structure, and in these helical structure, amino acids are joined together by the hydrogen bond if there are two polypeptide chains. In case of the secondary protein, if there are the two helical structure, two polypeptide chains, and the amino acids of these two polypeptide chains joined together by the hydrogen bond, so such structure is called as beta plated sheet. But the most common structure of the secondary protein is alpha helix. Right. So these are the certain characteristics of the secondary protein. Secondary protein has some structural roles. It plays structural role, and the examples of the secondary protein, because it plays the structural role, so it is present in the structural part of the plants and animals. And the examples of the secondary protein are the keratin here, and nail protein, and myosin. Myosin is a muscle protein. So you can see these proteins are involved in the formation of structures in different organisms. Right. Now the third part is tertiary proteins. The structure of the tertiary proteins is more complex than the secondary one. In this case, primary structure of the protein coil and shows further bending like so. Secondary structure, one secondary structure, secondary structure of the protein coils upon itself, and there is a third secondary structure. So secondary structure, they coils upon themselves and form a complex structure which is called the tertiary structure of protein. So this is more complex. And this structure is found in functional proteins. Functional proteins. And what are the functional proteins? For example, hemoglobin. Enzymes. So tertiary structure is more complex and it is found in the functional proteins like enzymes. And enzymes have the specific function. You know, all the enzymes have their own specific function. Like enzyme for the digestion has their specific function, and other enzymes for all other metabolic activities have their own role. So the tertiary structure is found in those proteins which have a specific function. Okay, there is another point. In case of the tertiary proteins, peptide bond is formed, hydrogen bond is formed, as in case of the secondary. And there are some additional bonds like ionic bond if protein is ionic in nature, and sulfide bond is present if amino acids contain the sulfur. So these bonds can be present in the tertiary protein. Okay. Now the last one is, last structural classification of the protein is quaternary structure. Or you can say quaternary proteins. Quaternary proteins are globular in shape and they have the 4 polypeptide chains. They are composed of 4 polypeptide chains in a complex manner, 2 alpha polypeptide chains and 2 beta polypeptide chains. And the example of the quaternary protein is hemoglobin. Hemoglobin is a complex type of the protein which is a quaternary protein, and same as in the bonds are present in a quaternary protein are those which are present in the tertiary proteins, like polypeptide, again hydrogen, ionic, as well as disulfide bond can also be present in the quaternary structure as in the tertiary structure. Okay, these all are the classification of the proteins on the basis of the various bases. So after listening this lecture, you can ask your questions